Hello, my name is Professor Matthew Schmidt, and I'd like to welcome you to Genetics. In this session, we're going to continue the theme of revisiting Mendel in the sense of looking at situations where it seems, or it apparently looks like, results that we've gotten don't really go along with what Mendel said. But as I think you're beginning to find out, Mendel's principles do always hold. So in this section, we're going to talk about gene interactions, epistasis, and modified Mendelian ratios. So this is a recognition that while, as you're going to see in a moment, sometimes when you do crosses, you come up with these ratios that look on the face of them nothing like they should if Mendel was correct. And again, we ask ourselves the question, wow, does this invalidate Mendel's rules in these organisms or under these circumstances? Or is there another way we can look at the whole thing so that it falls into the Mendelian scheme of things? And it's always true that it falls into the Mendelian scheme of things one way or another. So let's take a look at a couple of terms, and it's very important to understand not only the terminology, the name of it, but what we mean by it. So a very important term here is called epistasis. And epistasis often will, it's not the only thing, but it will often lead to these modified Mendelian ratios that we're going to be dealing with in a moment. Now the word epistasis means, or I should say epistasis happens when one gene interferes with the expression or action of another non-allelic gene. So before we go on, let's just make very sure we know what we mean by the term non-allelic gene. It basically just means this. For all of the traits we've looked at so far, the trait is controlled by one gene, or I could say one locus, right? If it's flower color, it's the P locus, where you can have big P or little p. If it's height, it's big T or little t. It's one gene that is affecting or determining that trait. With respect to epistasis, there are actually two different genes. If you were to draw it out letter-wise, in other words, one gene may be the A locus, where there could be the big A and the little a allele, just as an example, and then a totally different gene called B, a different locus where there could be the big B and the little b allele. We've seen this before. It's just that in the past, these two different non-allelic genes tend to control two different traits, like one was flower color and one was height. In this case, you still got those two separate non-allelic genes, but now they're somehow working together in a sense to determine one phenotype. So epistasis means particularly it's not an even sort of contribution, that one gene is sort of determining, controlling, interfering, however you want to say it, with the action of another gene. And between the two of them, we take both of what they're doing, we take what they're both doing into account. That's how we can figure out what's going on with the phenotype. So as I say here, it can produce all sorts of variations on the Mendelian theme, and we want to, this is sort of the most simple example of a general phenomenon referred to as multifactorial inheritance. In fact, usually when you hear multifactorial, the implications that sometimes there's 20 or 30 different genes involved in a trait, and also it can imply that there are some environmental aspects as well. In other words, it's not one simple factor. If you remember, I know I told you we weren't going to use this term again, but Mendel called the genes factors. So multifactorial means more than one gene, and even sometimes more than one gene plus some other influences going on. This is simple with respect to multifactorial. In fact, it's two factors acting here, but two is multi in the most simple form. So to be very clear, in the versions that we're looking at, this means that two different loci, two separate non-allelic genes, are involved in determining the phenotype of one trait. Now the two genes assort independently, the way they go through meiosis, etc. It's total independent assortment. So what we have to do is keep the genetic format of what Mendel was doing, but try and understand how the different ways the genes are acting are coming together to allow these ratios to occur. Now, one thing that you have to be clear about, if you're dealing with two loci, and if you did a cross in the form that we're used to doing it, in other words, we've often done a double, when we were talking about dihybrid crosses, we did a cross of the form big A, big B times itself, right? And we found that when we did that, because of independent assortment, we got a 9:3 to 3:1 ratio, right? So what I want you to very definitely keep in mind, memorization is important sometimes, but if you understand this, you'll be able to apply it to new situations, and often that's what you're asked to do. So the weird, if you will, ratios that we're about to see can always be understood as some variation of the 9:3 to 3:1 ratio, as long as we're doing the crosses in the same general format. Once you understand it, you'll be able to extend that to other formats. So, in a 9:3 to 3:1 ratio, just to be clear, genetically the 9, the 3, and the 3 and the 1 are, remember, the 9 are individuals in which there's at least one big A and one big B. In this case, complete normal dominance is what's taking place. It would get really complicated if we threw in some of the different dominance relationships that we've discussed. So for now, we're going to go back to just regular complete dominance. So 9 have one big A and one big B, and phenotypically they will look dominant for both traits. Remember, in this case, we're going to show that it's only one trait, but traditionally we're used to two. One of the 3s has one big A and two little b's. One of the 3s has two little a's and one big B. And the 1 is always homozygous recessive at both loci, right? Keep that general form in your mind, please. I beg you, okay? So let's look at one of the most interesting and, in my experience, prevalent types of crosses and questions that can be asked on an exam or even off an exam. So here's an example with sweet peas, and sweet peas are actually a different species than regular peas. So, but you can see why this would have been startling to the people who did this experiment. So we're going to do the experiment in a similar way as Mendel did, and what I mean by that is we've got in the parental generation, a true or pure breeding purple flowered plant and a true or pure breeding white flowered plant, right? Now, if this was the Mendelian world, I'm sure when they first did this experiment, they said, okay, well, it's probably just like regular peas. So this would mean big P, right? If it's pure breeding. This would mean little p, if that's pure breeding. Then, if the cross, the F1, would be all purple flowered plants, and that's what they saw. So far everything was smooth sailing. What would the genotype of all these be? They'd all be big P, little p, right? They're heterozygous. So far everything's, as I say here, as expected. But it's in the F2. Remember, the F2 is we're going to cross these two. And by the way, I'm writing these genotypes in. They're not correct. It's as if we were thinking of a Mendelian model, this is what we'd be expecting. And what would we be expecting in the F2? Wouldn't we be expecting that traditional 3:1 ratio of purple to white? Sure, I mean, you don't have to, I think, by now, but you do a little Punnett square. If these were the genotypes, we would get a 3:1 ratio. But it turns out, look what we get. We get, in the F2, a 9:7 ratio of purple, 9 purple to 7 white. Now, you can do all the mathematical manipulation you want. 9:7 is never going to turn into 3:1. They're just completely different. And yet, they did this over and over again and determined that it really is a 9:7 ratio. You can't even reduce 9:7. 9/16 are one way, purple, and 7/16 are another way. How can we possibly explain this? Well, I wouldn't dare to say that it's easy, but I think once you get the hang of it, you'll understand exactly what's going on here, and it will not be totally mysterious, I hope at least. Because here's an explanation model. Now, when I say it's a model, in reality, the way these genes interact is not quite as important as being able to understand to understand why this type of a ratio occurs. So I'm going to give a little bit of an oversimplified model, but it's good enough for you to understand. And then I'll show you another model which is probably more close to the truth. But here's the explanation. So what we're going to say, as you might have guessed, is that flower color in the sweet pea, for whatever reason, is not controlled by one single gene or one single locus. That two loci are involved. Another way of saying that is two non-allelic genes are involved in this one trait, pigment production, in the sweet pea. So you may see these loci called different letters. That really doesn't matter that much. But I like to use C and P. So here are the two loci that are involved. The C locus here, I want you to think of that as a switch. It determines whether or not any pigment will be made. Now the big C allele is dominant to the little c allele, and the big C means some color is going to be produced. I think of this as a switch. It basically, if you got a big C, the switch is on and it says go ahead and make your pigment. If the switch is turned off, that's little c, no pigment's going to get produced. All right? Now the P locus actually acts very similarly to the P locus you're used to in regular peas, in that big P will give a purple flower color and little p will give a white flower color. But you have to put it together. So if you look back at our cross, what really happened, not what I wrote down, which was a assumption that it was going to be a simple Mendelian cross, the F1 really was big C, big P, little p. It wasn't just one heterozygote times another. It's a double heterozygote because two loci were involved from the beginning. Just very quickly, I don't want to go back to that page, but the original pure breeding plant was like this. That was the purple one. The white one was like that, and therefore all of the F1 were like this. Okay, and that's the cross that we're illustrating here. Now, whenever you do this, as long as independent assortment is happening, which it is, you get a 9:3 to 3:1 ratio of the form that we said. So this part right here should not be very shocking. 9, which are going to have at least one capital C and one capital P. 3 that have a capital C but are homozygous for little p. 3 that are homozygous for little c and have at least one big P, and that 1 that's always doubly homozygous for the recessive trait. But now here's the whole key to this, you guys. If you put together, there's two different genes, and the C locus is epistatic to P, meaning it sort of interferes with P. Think of it this way. P is basically saying, I'm going to make purple color, I'm going to make white color. C basically says, well, I'm in control of that. I'll determine whether you do anything or whether we don't hear from you at all. So think of it this way, please. If you have a big C and a big P, what that can be interpreted as saying is, big C means, okay, I'm going to flip the switch, make your color, go ahead, I'm allowing that. And therefore, if there is also a big P there, the color that's going to be made is purple. So this group of 9/16 are going to be purple, right? But look what happens with the other groups because of this epistasis. This group here, get a little different color going, the big C gene is there, allele I should say. So that means it's, again, saying flip the switch on, go ahead and make your color. But what color gets made? Well, at the color locus, or I should say at the pigmentation locus, you got little p. That's a white flower, right? So this group is going to look white. For a different reason, this group is also going to look white. What's the reason? Because even though there's a big P there, the epistatic C locus is little c. That means we're shutting down the switch. I don't care what you have over there with your P's. You're not making any pigment, and that's looking white as well. And in this case, it's sort of a double whammy because the switch is off, and even if it were on, you'd be making white coloration. So while genetically different, the 3:3:1 part of the 9:3:3:1 ratio, all this one, and this one, they all look white. And that's 3 plus 1 is 7. So you end up with this 9-to-7 ratio. Another way of saying that is you could do a Punnett square, and I'll actually show you one in a minute, but there are 16 possibilities when you do a dihybrid cross, correct? And in this case, 9/16 of the offspring will turn out to be purple, while 7/16, again, not always for the same reason, but phenotypically they'll turn out to be white. Why don't we take a look at that Punnett square just to drive this idea home? Now, purposely I did this because you'll see this, once you understand this principle, that's awesome, obviously. But sometimes then questions will be asked to you, which even if you understand this, you can still come up with the wrong answer, unfortunately, if you don't apply the information correctly. So randomly, sort of, well, it was planned out, but I just mean you could do this across a bunch of ways. But I want you to see something. The way this is, now we happen to use A and B here just to make it of a more general format, but AA, big A, little b, would be the same as what we called big C, little p, and this would be the opposite. Very interestingly, if you understand the model, both of those white flowers that you're starting off with are pure breeding lines, right? Because this one will always have big C and little p there. And as long as you have two little p's, you're going to be white. This one will always have little c and big P. It'll still be white for a different reason because the switch is shut off, right? But this is why it's interesting. And this can confuse you, but not if you keep in mind and be very methodical about it. Because the genetics is the same. And this is the evidence that Mendel was right. So all of the F1 in this type of a cross are going to be big C, big P, little p, or in a more general way, whatever letters you decide to use, big A, big B, little b. Now remember, to be purple under this model, all you need is one big C and one big P, and the F1 have that. So all of a sudden, I don't think we've seen this before, we had two white flowered plants that were pure breeding, in fact. We crossed them, and now we've gotten a purple F1. This is really one of the few ways to explain that. Certainly Mendel never saw that in his crosses. Now, what he did do, though, if you cross these double heterozygotes of the F1 with themselves or with their brothers and sisters, you will get a Punnett square. I don't know if we have to go through the whole entire idea of the Punnett square. Just remember, since they're double heterozygotes, each of these parents can make 4 different gametes. And what you want to do is just to understand this aspect of it is any box where there's, well, in my language, at least one big C and one big P, you're going to have purple, right? What that means as it's written is any box where you see one big A and one big B at the same time, then that's going to be purple. And it's colored in very nicely. So 1, 2, 3, 4, 5, 6, 7, 8, 9. If you take a little closer look at that, if you want to pause, every one of those and only those has both a big A and a big B in it. All the ones that are white, if you really wanted to do this, you'd find 3 of them that have big A, at least one big A, but they're homozygous for the little B's. Change colors. You'll find 3 that have at least one big B and they're homozygous for the little A's. And then you find that one that's homozygous recessive at both locations. But look-wise, all those 3 to 3 to 1 look white, right? So this shows you, at the risk of redundancy, the genetics of this is a pure dihybrid cross. What's different is the way that these two loci are interacting to affect the expression of one, what we've called a trait, or one phenotypic trait, purple or white. So this 9-to-7 ratio is a modification of 9 to 3 to 3 to 1, and basically all of these 7 are looking the same. And these are looking purple. So there's your 9-to-7 right there. Believe it or not, this is actually fun. You do a little detective work and say, wow, that makes sense. Now, there are a variety of types of questions that could be asked in this situation, and you saw one of them. Don't make the mistake of thinking that you could have purple pure breeding line. If you had a purple pure breeding line, I hope you can see that it would have to be like this, because all the offspring would always have at least would have two big C's and two big P's all the time. But realize you can see it right here. There are two different ways that you could get a white pure breeding line. And I should add, there are three different ways, because this down here would be pure breeding as well if it's doubly recessive at both loci. So these are the type of things that you have to keep in mind, and that can unfortunately trip you up possibly when you go to solve problems that are presented to you. We'll talk more about this in the problem-solving sessions, but the key is to try and establish what you're working with before you jump right into things and start, doing a problem and then realize that you've done it wrong. So there should usually be some clue if it's this type of a situation that something's amiss. Either they'll tell you it's a 9-to-7 ratio, or you'll see something like two white plants making a purple like you see here. So make sure you're comfortable with this, because all the rest of the things we're going to do in this session are basically variants of this type of a situation. Now, here's a biochemical model for purple pigment production, which is not exactly what I said, but it would explain it exactly the same way. So think of it this way. What if there's a colorless precursor, which is a nutrient perhaps that the plant picks up from the environment? And what if allele A, or whatever you wanted to call it, P or C, changes this colorless precursor via an enzyme that allele encodes into a second precursor, which also happens to be colorless? And then you'll have allele B, which makes another enzyme that works directly on this and leads to purple pigment, and there is a purple sweet pea flower. The bottom line, whichever model you look at, is you need at least the capital letter, the working allele for both of these, to make the purple pigment. If you're missing either one of them, it's going to be white. And so even if you look at it this way, whether you're missing that enzyme, you can't complete the pathway. If you have that enzyme and you're missing this one, you can't complete the pathway. If you don't have either of them, you can't complete the pathway. So this is just a different way of looking at it. This may be more biochemically correct, but usually you don't have to worry about it. So let's take a look at some other examples, and really, if you understand the first one, the rest of these I think are not going to be so bad. So coat color in rodents. We're going away from the plants and getting into animals. So here's a 9-to-3-to-4 ratio. Now, if you're astute, and I know you guys are, you can see right off the bat that 9-to-3-to-4 is sort of taking the 9-to-7 and breaking it out a little bit more, right? It still always adds up to 16. So in this case, it's sort of similar to what we saw before. One locus determines the presence or absence of pigment, like that switch, just like before. Big C means turn it on, let's see what you got. Little c means shut it off. The other one determines what color will be made. This is very similar to the situation we just saw, except for one thing. The big A allele gives a color called agouti. This is another one of these situations where there's an actual animal called an agouti, but it also refers to a coloration pattern in various creatures, in particular rodents. So in this case, you see an agouti mouse there. It sort of looks like a tannish color. For now, don't worry about it much more than that. But the little a allele makes a black pigment. So see how this affects things. It's a little bit of a different biochemical pathway. I don't think we need to go through every time that if we do a cross, what I'm assuming here though, let's just be clear, is that we're crossing an F1 that's doubly heterozygous. That's when you get the 9-to-3 to 3-to-1 ratio, right? But we don't have to draw the Punnett square every single time. So look at what's happening here. As long as there's a big C around, it's saying switch is on, go ahead and make your color. But if there's a big A, the color that gets made is agouti. If there's two little a's, the color that gets made is black. On the other hand, if you have two little c's, the switch is off and you're making no pigment, which pretty much equals white, but in the case of animals, they refer to this as albino. So both this one and this one are albino. It would have been the same as before if this made a white pigment, right? But we're throwing another pigment into the mix. So what we've got here is this one and this one look the same. So that's the 4, and you have 9 to 4 to 3 in your ratio. 9 agouti colored, 4 albinos, and 3 black. I hope you can see that it's very similar to what we just did, but it depends how the genes are interacting and what it is they're saying to each other. Mendel knew nothing about this, but that's almost the beauty of it. His principles still are holding when we discover more and more and more. The 9 to 3 to 4 ratio. This is exciting stuff. It really is. Let's just take another one. You could do this all day long, and I'm sure you'd like to, but we don't have all day. We could do a few more, though. So fruit color in the summer squash. I just want to say here are some beautiful pictures of some yellow summer squash. The word squash, and pumpkins, and gourds, and all this stuff, they're all very related species. Sometimes they're even the same species. So if you've heard, it may even depend where you live, what these things are called. But the summer squash is a commonly eaten, it's a fruit. I don't know if you think of it as one. But in summer squash, when we're talking about the color, now we get a 12 to 3 to 1 ratio. I hope you're picking up that every one of these adds up to 16, and not only does it add up to 16, there's got to be a way of fitting it into the 9 to 3 to 3 to 1 ratio scenario, right? So again, in this case, look what's happening. One locus, which we're going to call W, somehow if there's a big W there, it doesn't matter what else is going on. White pigment's getting made, and that's it. If it's little w, it means not white. Whatever else the other locus says to make is going to get done. So if the color's not going to be white, the Y locus, big Y is dominant, which is yellow, to little y, which is green. So there are 3 possible colors, white, green, and yellow. Again, I really want to make sure that you understand this. So the cross we're doing to get the 9 to 3 to 3 to 1 ratio is this one. If they establish, for example, that this is what's going on and then give you a different cross, it only gets easier, but you won't see the 12 to 3 to 1 ratio. You just have to use your logic about what goes on in this type of a ratio. What I mean is, so in this case, you're told, well, you may or may not be told it depending on the extent to which your professor makes you memorize it. But once you see 12 3 to 1, you know what's happening. Because wherever there's a big W, you're getting white. It's totally irrelevant what's over there. And so there's the 12 right there. If you have situation with little w, what does that mean? It means the white gene is not asserting itself, if you want to think of it that way. It's saying, whatever, I'm not getting involved with this. So now I defer to the yellow locus. See how that's epistasis? One is it controlling or interfering with the other. It's almost as if in these cases here, the yellow locus is saying, hey, I want to make yellow color. And the white locus says, I don't care. I'm making white and you can't do anything about it. But when it's little w, like it is over here, as long as there's one big Y, you're going to get yellow. And there are 3 of those. And if there's little y, you're going to get green, which is the recessive. So there's clearly why you get the 12 to 3 to 1 ratio. I hope this is sinking in with you guys. I really do, because it of course depends on the class, but sometimes some large sort of essay style problems could be related to this type of phenomenon. It could drive you nuts if you don't see what's happening. So if I were you, I'd always look for, if you just have no idea what's going on, but you have a feeling it has something to do with this, think 9, 3, 3, and 1, and see how what they're giving you might fit into that type of a ratio. Okay, so in this case, let's look at a Punnett square just to make sure we understand what's going on here. The B locus is epistatic to the A locus. Again, we did it here just more generally using A and B. In this case, the B locus would be the same one as what we call W. So B equals W, A equals Y. I apologize, but sometimes you'll see it written just more generically this way. So again, there are a lot of ways you could draw these crosses, but in this case, we have a pure breeding white plant, and it's pure breeding because what do you need to be white? In this case, all you need, because remember, this is the B, all you need is a capital B, and that's it. You're white. But this is pure breeding because whether it was big A, big B, or little a, big B, it's pure breeding for white. The only way to be pure breeding for green is little a, little b, because that's the only way to be green anyways, right? But as you suspect, the F1 are double heterozygotes, and as long as there's one big B, it's going to be white, right? So it's so interesting. You cross a white with a green, you get two whites. If you didn't know anything else, you'd say, oh, it's a simple Mendelian thing. White's dominant to green. But then you do the F2 based on this cross, and all of a sudden you start seeing whites and yellows and greens. And the reason is, I know you now understand it. Anything with the big B allele, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, they're going to be white. It supersedes anything else. The four that are remaining, little b is homozygous for little b is basically saying, whatever, I don't care about the color anymore. You decide A locus or Y locus, as we looked at it before. So if it's got at least one big A here, big Y, it's going to look yellow. There's three of those, right? 1, 2, 3. And we get our one that's green. So you think you cross a green with a white, all the F1 are white, the F2 have a 12 to 3 to 1 ratio of white to yellow to green. It's really the only way to explain it. And some people don't, but some people love trying to put this type of stuff into exams or just into whatever assignments you're going to get. So it really does pay to understand it, unless of course you know for a fact that this is not so much on the table, but likely it is. And understanding this just really deepens and enhances your understanding of genetics in general. So that you see that one principle can just be expanded upon into all these seemingly different possibilities. All right, where do we want to go from here? Well, some of you may want to go and get some coffee or something. And while that's not a bad idea, if you want to do it, pause it first. I can't do that, however, so I'm going to keep going right through. So I'm going to give you two more examples. There are more of those other types that involve epistasis. I'm going to show you two rather quickly that do not involve epistasis but still involve gene interactions. Epistasis implies that one gene is sort of interfering with the other's activities, right? Well, here we have a situation where other types of gene interactions still produce 9 to 3 to 3 to 1 ratios because the cross is always in the same form. But depending what these alleles do and their relationship with one another, we're going to see different. In this case, we're not going to see so much strange ratios. We're going to see our 9 to 3 to 3 to 1 ratio, at least in this case. But instead of having this, it's affecting one trait, but in a way that's different. I'm sorry, I know that wasn't the best, but you'll see what I mean in a minute. So for example, in chickens, two non-allelic genes interact, just like before, but now it's in a non-epistatic way, meaning one isn't really stopping the other or whatever. They're combining in a way that we don't have to understand biochemically to both contribute, not in an antagonistic way, but in a cooperative way, if you will, to determine the comb type. The comb is that thing in chickens that sort of hangs down from their chin, I guess. Do they have chins? There are four possible phenotypes: the walnut comb, the rose comb, the pea comb, and the single comb. So the bottom line is, I don't even know if it's understood why this happens, but it's fully understood that it does happen. In fact, let's just look at this one and this one for a minute. If you have one big R and two little P's, you get the rose comb, and that's what the R allele was named after. If it's the opposite situation and you have a big P and two little R's, you get a so-called pea comb. I don't know who came up with these names. But check it out, just as an example. If you crossed, let's do this. If you crossed a rose comb that was true breeding times a pea comb that was true breeding, wouldn't all of your F1 be like this? And guess what? That's having a big R and a big P. That's the 9, and that gives you a so-called walnut comb. So it's another one of those weird situations where, in this case, you're getting there are 4 different phenotypes. So you cross a rose with a P, you get walnut. That seems odd. It takes a while to work this stuff out. But if you understand the idea, just do that Punnett square or do it however you want to do it. Get the ratio, get the results, and then based on what you know about how these alleles either interact with each other or combine to form a trait, you could figure it out. I'm telling you. Now, if you're the 1 and you have small letters at all locations, they call that a single comb. And this is an example. So in this case, the ratio is unchanged. This isn't a modified ratio like 9:7 or 9:3:4. What's happening though is, even though it's 9:3 to 3:1, each one of those has a unique phenotype associated with it, and that's common when you have non-epistatically interacting pairs of genes. Okay? So whatever those gene interactions are, they're leading to these different phenotypes, and usually you have to be told that, unless you're told to memorize it, like I happen to remember, but to know what's a walnut comb and what's a rose comb, many times they would tell you in the problem, and then you would have to apply what you knew. But better safe than sorry. So one last one. Here's another non-epistatic situation, and it does alter both the ratio, but you'll see why it is sort of related to the idea of with the chickens over there, even though we're back to summer squash. Now, again, this is not a botany class, but the idea of a squash or a gourd, I don't know where you guys live. I live on Long Island, New York, which is a beautiful place, mostly. But in the autumn, as I suspect happens a lot of places, you go out and buy pumpkins, which actually is a type of squash if you want to go that way. But there's also these little flattened things that sort of look like pumpkins. They're mainly decorative, and people call them gourds. Let's just say pretty much these are, if they're not the same species, they're all very closely related type of plants. So I'm just going to keep it with the summer squash, but we have 2 non-allelic genes. They're also interacting non-epistatically to determine the fruit shape. So there's a few shapes. I don't have pictures, but the disc shape is sort of a flattened one. It looks sort of like a gourd that I had on my desk once. I'm a terrible artist. I guess you guys have figured that out. Sphere obviously is more, like an orange or something. And the long shape, well, the long shape, if we want to go back, that's this right here. That's what they call the long shape. So 3 possible phenotypes, it turns out, are possible. And here's another. I mean, you may be saying, this is overwhelming. How can I remember? What you need to remember is that the products of different genes act in various ways to combine, to interfere, to do whatever it is they're doing to determine the ultimate phenotype. In this case, it would appear that an enzyme probably made by the A allele functions very similarly to one made by the big B allele. And the reason I say that is because whether you have a big A or a big B, but not both, those all look sphere-shaped. So that's where you get the 6 in this ratio. If you have an A and a B, there's an implication that there's a double dose of this activity, and that's not clear why, but that makes the disk shape. So we've got 9 disk to 6 spheres, and only when none of these enzymes are present, you get the long shape. So it's 9 to 6 to 1. Now, I hope that that's clear. It could have been, I'm not saying this works exactly like the chickens. This is an instance where the two enzymes are probably similar. Here's a thought question. Which fruit shape is always true breeding? Now, don't get me wrong. You could make true breeding lines of any of these and probably in a couple of different ways, right? In other words, clearly, this and this would both be pure breeding for the sphere shape, right? But you can have a sphere shape like this, that is not pure breeding. Do the Punnett square and you'll see. So which one is always true breeding? I think you guys have got it. It's got to be, which one? The long shape is the one that is always going to be true breeding, right? Because the only way to be long is to be little a, little b, and that has to be true breeding. If you go through all the other ones, they can be, but there are situations in which you could be heterozygous at either locus, and then you're not going to be true breeding. And I really love this session, and I love this idea. I understand if you don't necessarily love it, perhaps you'll come to love it, but it's so important to understand this stuff. So I'll give you a while to digest it, and I'll see you again real soon.